This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 192, recorded on January 3rd, 2019. Happy New Year, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How's your year so far?
It's pretty good. Yesterday, I turned 66.
Hey, congratulations.
Oh,
I am
a youngster.
I am trying to catch up to you, Elio.
Well, you got a ways to go.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, and happy birthday to you, Vincent.
Thank you. And from Charleston, South Carolina, Michael Schmidt.
And Happy New Year as well to everyone.
Yes, Happy New Year. First TWiM of 2019. And this year we will reach our 200th episode.
Isn't that nice?
That's cool. Another year of TWiMing. We'll be at ASM Microbe. And who knows where else.
That's true. We haven't taken ourselves on the road very much. No. You go off on the road, but...
Oh, I have to tell you, TWiM did a lot of road shows last year. I think...
TWiV.
I think we did 14 or so. And I had a T-shirt made, a TWiV World Tour T-shirt with all the dates on the back and the places we were...
Oh, that's cool.
I had actually a virologist. She is a good artist down at Florida Gulf Coast University, Sharon Eastern. She made it for us. And I just ordered one. Looking forward to that. So someday we'll have a TWiM World Tour.
A world tour.
Yeah, one show a year does not constitute a world tour. But TWiV was actually in Europe a couple of times last year, so it really is a world tour. It's not every continent, but in an effort to make ASM's podcast the best that they can be, This Week in Microbiology wants to learn more about you, the listeners of TWiM. Please take 5 minutes to answer a short 12-question survey. It'll help us collect the data we need to seek sponsorship of the show. The survey is at asm.org/TWIMPoll. That's asm.org/TWIMPOLL. One word. Thanks for your time and listening, subscribing, and helping us improve the show. We appreciate the support. From Anthony comes a link to vote for the New Jersey State Microbe. Put a link in the show notes. The state microbe, there's a push to make Streptomyces griseus the official state microbe of New Jersey.
And can you give us a hint why that would be?
Well, that was discovered by Selman Waxman and his colleagues at Rutgers University and has led to many antimicrobials.
All right.
Streptomycin, for example. And so
big one.
John Warhol, who was on TWiM a couple of months ago, has been the big push for this. Anyway, there's a website at change.org. 638 people have signed out of 1,000. I don't know what happens when you get to 1,000. Then, but anyway, New Jersey should have one. Wisconsin has one. And
must be a lactobacillus, I'll bet.
It's a cheese producer, right?
Yep.
Mm.
Yep.
And also, I think Oregon has one for beer.
Well, that would be a yeast.
Yeah. Let's look it up. Oregon State Microbe. Yeah, it's Saccharomyces cerevisiae. Oregon was the first with a state microbe. Let's see, Wisconsin State Microbe. Lactococcus lactis.
Yep, making cheese.
Does Michigan have one, Michele?
Not to my knowledge, but I could be missing it.
Let's look it up. Michigan State Microbe. No, nope.
That would be Victorita, the Michigan State microbe.
Oh, look, there's a Hawaii Flavobacterium acane vivens and Alli vibrio fisheri. Hawaii, yes, Hawaii. It's pending. I guess the Lactococcus is proposed also. So maybe only Oregon has a past one. Now, there's a Wikipedia page for all this. How about that? State microbes. Why not make microbes famous or more famous, right?
Mm-hmm.
Give them their due.
They should have their due. And we're a big fan here on TWiM of microbes in general, so it's a good thing to do. All right, let's go on to some papers, the literature. And Elio has the first snippet of 2019. What an honor.
Okay, I'm looking at a highway sign that says entering Mastodon Township, home of the humongous mushroom. This is a place on the Upper Peninsula of Michigan where in a forest is a giant mushroom in the sense that you can find specimens of a mushroom called Armillaria gallica. I'll talk about this in a minute. When you look at them, you find that they're all identical. They're all part of one single clone. Now, mushrooms can do that because they are the product of, they're the fruit of an organism which is found underground over vast areas and which is composed of filaments. Each filament is called a hypha. I like to say that's not a port in Israel. It's something else. The filaments which together are called a mycelium. So a mycelium can be the size of a county, and in the one in Michigan is said to be one of the largest things on Earth, and it covers some 90 acres of a forest. It is estimated to be over 100 tons in mass and about 1,500 years old. That's the old data. Now, let me tell you that anybody can claim bragging rights, and us mushroom lovers can claim that a mushroom is the largest organism on Earth, but it ain't so. The reason is that aspen trees do the same thing. They send out roots, and from the roots comes a new tree, so that the whole forest of aspens is gigantic. There's one in Utah, which is 106 acres large, called the Pando. So,
well, speaking of bragging rights, may I interject that the northern Michigan, the Upper Peninsula, is the land of my forebears.
Mm-hmm.
So this is western Upper Peninsula. It's due north of Green Bay, Iron County, I believe. Mm-hmm. And they called it the humongous fungus.
That's right. It is a mushroomissimo, you could call it. Anyhow, so the story is that this mushroom has, or this organism, first of all, let me say something about Armillaria. Armillaria is also known as the honey mushroom because of the color of the caps. It's a very good eating mushroom. There's some preference in some countries. In parts of Italy, it's considered to be great. Here in the United States, we think it's okay. It's an okay mushroom. So it goes from okay to delicious. Anyhow, it's a plant pathogen, a tree pathogen. It can affect live trees and kill them. And it also grows on dying trees a lot. And you can find, when you find it, you can find a lot of it because you'll find a collection of maybe 50 caps in one place. Wow. So for those who like it a lot, it's a wonderful thing to find. So Armillaria gallica can be humongous. Now, the interesting thing is that they went back, the original authors went back. Oh, by the way, in Oregon, there's a bigger one, 3 square miles, and maybe 8,000 years old. And so that's probably the biggest one. But the one in Michigan is big enough. And the people who studied this found that there are a couple of interesting things about it. This Armillaria has unusually large genomes. And the idea is that maybe it's to accommodate purported virulence factors. We don't really know what the virulence factors are, but they must be there. And maybe they take up a lot of DNA and make the genome extra large. The other thing which is only interesting to us mushroom lovers is that when a mushroom is made up of a filament, as I say, the filament comes up when spores germinate, and they have to be of a different mating type. When this happens, different mating type hyphae will fuse, and something happens which is very strange. The two nuclei do not fuse like they do in our cells. Okay, so that instead of having a diploid, you have a dikaryon, a heterokaryon, if the nuclei are different. And they stay that way until just before, when they make a mushroom, this is the sexual stage of the organism. At that point, they undergo karyogamy, that is, the two nuclei fuse, and then they meiose and give you haplospores. Anyhow, that's to those of us who care, this armillaria is unusual in a couple of different ways. Anyhow, another thing I should say is that armillaria can crawl all over the place because their hyphae fuse together into long filaments called rhizomorphs, which means root-like shapes. They're also known as mycelial cords, which can anastomose and cover a huge area. So the story of the giant mushroom has to do with their ability to make these underground structures, which can go. They grow, by the way, the speed of 1 to 3 feet a year, which is significant. And they are foraging agents because they find new potential hosts. So this is a highly successful parasite. So returning 25 years after discovering the Mushroomissimo in Michigan, they returned to the site, and this time they had more powerful genetic tools. So they found that their friend was still there. It turned out to be 4 times larger than they had measured originally, and older, about 4 times older. Anyhow, using more detailed genetics, which included determining the whole sequence of a bunch of mushrooms, they studied essentially 245 samples and measured the growth rate of the rhizomorphs, these underground structures. They sequenced actually 15 samples, and they found that out of a genome of 100 million bases, just 163 point mutations.
Wow.
That's nothing, right? And they figured out the mutation rate is about 10 to the minus 10th per base per cell replication. E. coli is said to have 10 to the minus 6. So it's orders of magnitude more stable. So they expect a mutation every 50 cell divisions, which is essentially nothing. So here you have an organism which grows and grows and grows and grows and lives for centuries and doesn't change very much. So what goes into this? Well, the paper, which will be in the show notes, gives a fair amount of speculation, and it really they go into a lot of length. I won't do that here because the speculations are interesting, but beyond what we can do in a short time. But they involve peculiarities of the rhizomorphs, which may be one reason. They also say it could be a highly efficient repair mechanism, which doesn't quite sound right, and other things. So there are a lot of things that picture this. So anyhow, that's pretty much the story. The genetic stability is very interesting, and they make an interesting point in their conclusion. If I can read it, let me read it. Our picture of clonal evolution in individuals with armillaria closely parallels that in cancer progression within single individuals. Cancer progression, however, is accompanied by extreme genetic instability rather than the loss of control over DNA replication. Whereas here, I'm stopping the quote, here obviously it's the opposite. So cancer is one extreme of an enormously high genetic instability. This is the extreme of, this is a sample of enormous stability. So they end up saying the genomic stability of armillaria and the underlying mechanisms allowing such stability may provide a useful counterpart to cancer. I'm not sure that people in the cancer field are going to feel that this is of much use to them, but it's the opposite. So if you like opposites, this is what you have. So that's about the story.
I guess we can't judge whether it'll be useful to cancer or maybe designing new therapeutics until we know the mechanism of that stability.
That's right. Yep.
I think you're right. But, it's nice to bring up interesting sounding
For sure.
ideas. It's
Yeah.
an idea.
I mean, I guess part of the stability is that it replicates very slowly, right?
Well, it doesn't replicate all that slowly. I mean, it grows fairly fast.
What is it, a couple of feet a year?
That's, in length.
So what's
Right.
so how often will it divide? Do they say?
No, they don't know. Well, actually, no, they do know. I don't know. I forgot. I don't remember what they say.
What's so surprising is we think of kind of a background level of mutation as being beneficial because then some members of the population can adapt. So it suggests that the environment there is very stable, perhaps, or
That's right.
that
In fact, they make that point that a forest is not full of mutagens. They're not exposed to UV light. The chemical composition of a forest is relatively stable. So you're right. That may be possible reason why not much happens.
It's cold up there. They get a lot of snow.
So that'll slow anybody down. So the question then becomes is, as we're undergoing a period of rapid climate change in the geological sense, I mean, we've seen a rapid increase in CO2 over the last 100 years. You wonder whether or not something that does not have the ability to mutate, if you will, in real time, is going to have trouble adapting to the changing conditions it may encounter, because unlike mammals that can get up and move to a different location, this thing is effectively, for lack of a better word, landlocked.
Yeah,
that's right.
Well,
it's a good point.
It is a good point. However, as you know, the general change in climate has selected for fungi that can grow at higher temperatures, right?
Yes.
And their pathogenicity is increasing as a consequence.
But most mushrooms don't like high temperatures.
Yeah.
So there may be big changes. It'd be interesting to go back into another 25 years. If they're still alive, they probably will do that.
So, Elio,
or have the grant funding.
They say they don't know the number of cell divisions between any two isolates.
I don't know.
Yeah, so they don't really know. What I did find cool is that they had sampled a number of different individuals, right? So so that's a nice validation of their ability to distinguish, right?
Right. It's kind of weird, you go into a forest, you find a lot of mushrooms.
Yeah.
And then you decide that to analyze them and you find out they're all the same.
That's right.
That's surprising.
One idea that they put forth, which is really interesting, is that the template strand of DNA they postulate may be the one that
retained
doesn't basically is the root, and the new strand is kind of an offshoot. And so if the offshoot doesn't make it because of the mutation, that's okay. So assuming that the new mutations happen during replication, and as long as you put the newly replicated strand as an offshoot, then you're protecting the parent genome, I guess, is the idea.
You can do experiments in real time, and if it's deleterious, that arm effectively withers and dies off. Whereas if it's good, then it can effectively continue on with its path forward
and become stronger, maybe, and more fit. Interesting.
Well, this paper reminded me of the popular movie Avatar. Those of you who remember Avatar, and it talks about the symbiosis between hosts and plants and communicating, and how this giant tree on the planet Pandora was effectively doing it, akin to what I think Elio mentioned earlier, the aspen population. But when I first read this, I immediately recalled the plot of Avatar, and this is really what is actually going on in this. The genome is spreading literally across Upper Michigan, and it's, if you think about it, they have a similar founding species in Oregon, and it's really fascinating. And some of the crazy Hollywood plots do actually have some basis in real science.
Sometimes, but they
sometimes
sometimes bend it a bit, right, to make it interesting for them.
There's definitely science fiction bending.
Elio, if you go outside and see a mushroom growing, how big is the mycelium underneath that typically?
It's very hard to know because it's, how are you going to find out? You have to tease out the mycelium filament by filament. But most people think it's a question of yards, not of acres.
Right.
Something like that.
I know sometimes you see big circles of mushrooms, right? Like
you see
20,
fairy rings.
Fairy rings, right.
And they can be as big as, 10 yards, 20 yards, something like that.
So they're all linked by mycelia underneath.
They're all linked by mycelium underneath.
That's very cool. Very cool. I wonder if they're transcontinental growths. Probably not, right?
Why not?
They probably won't respect the wall. If there's a wall between Michigan and Canada, they probably won't respect it. I didn't say that.
The oceans. I'm thinking, no, Canada and the U.S. or Mexico, I could see it growing there, but the oceans would prevent going to another continent.
But what you need on land is to have the same habitat over a vast area.
Yeah, yeah.
This particular fungus is not going to be able to make it into Canada because Lake Superior is to its north.
Mm-hmm.
Yes.
Yeah, it's but eventually it will get bigger and bigger because it goes a couple feet a year.
That's right.
It'll be a long time. Oh, that's cool. Good story. Thank you, Elio.
My pleasure.
I love it. All right, we have a totally different paper for our main paper, and this is an article in Cell. It is called "A Host-Produced Quorum Sensing Autoinducer Controls a Phage Lysis Lysogeny Decision." And this is really unusual. There are just two authors on this paper, Justin Silp and Bonnie Bassler from Princeton University.
Kind of amazing, isn't it?
In the old days, you did this. My two postdoc papers were two people, but nowadays it's very rare because you have bigger groups of people and things are more complicated. But I always tell people if you can do something on your own, it's also useful to do something on your own. It's great to collaborate and learn how to collaborate, but doing something by yourself is also useful and very satisfying, and that's what we have here. It's pretty cool.
Well, I will jump in, and I did have a chat with Justin this morning, and he credits a postdoc in the lab who did the paper that he and the postdoc together did an earlier paper, and that was really the birth of this one. So Justin is too modest to say that he did this on his own.
What is Justin's position in the lab?
He's a senior graduate student.
Wow, impressive.
Yeah.
Well, this paper builds, as you will see, on a lot of phage genetics that has gone from mainly from Lambda, and it's just a classic example of classical bacterial genetics combined with modern technology. And I hope that I can do it justice because there are just a lot of experiments here, and I have to summarize some of them in the interest of time. But it's really quite remarkable, and I depend on my co-hosts here to help me out
on the straight and narrow.
So this is all about cell communication, the process in bacteria called quorum sensing, which is used to coordinate how the group behaves. And this depends on the release of signal molecules. They're called autoinducers or AIs. And it's been very well studied in Vibrio by the Bassler Laboratory and others. And Vibrio cholera in particular is well studied because quorum sensing regulates the genes that are involved in the human disease cholera. And the laboratory has recently found a new quorum sensing circuit in Vibrio, which consists of a cytoplasmic receptor transcription factor. It's called VQMA. I'm going to be saying that a lot. So that's a cytosolic factor that binds the AI DPO, which is dimethyl pyrazinol. We'll just call it DPO, small molecule that binds this transcription factor. Upon binding of the small molecule, you get transcription of a target gene. In this case, it's VQMR, and that encodes a small RNA that represses the genes needed for biofilm formation and the production of virulence factors.
Can you stop for a second?
I can.
This is kind of peculiar because most quorum sensing in pathogens lead to an increased production of virulence factors. This decreases the production of virulence factors, as if the bug says, there's enough of us, let's not do more harm.
And it activates their dispersal program,
that's right.
which
that's right.
Imagining is motility, so that now they can go out and seek ritual.
Virulence is a very difficult concept to talk about in a single word because it's not just doing damage. It's also letting the organism survive and the bug survive in the organism, etc.
It's a word used to describe behavior of the microbe.
That's right.
The other part of this paper is are bacteriophages, and we know in cholera that a phage actually regulates the virulence of the cholera because there's a major virulence factor, CTX, which is actually encoded in the genome of a lysogenic phage,
which stands for cholera toxin.
Cholera toxin, and they say something which I really like. Although Vibrio cholera is considered the causative agent of cholera, the etiology must also include the CTX phage.
You can't have one without the other.
The other part of this story is Lambda, which was worked on for many years. And some labs still work on Lambda. And what we know about lytic versus lysogenic reproductive phases. So some phages can infect bacteria and lyse them, but others are temperate phages. They can lysogenize the host cell and remain dormant. And the name prophage was first coined to describe that. And the initial work on Lambda integrates into the DNA of the bacterium. And that whole mechanism really gave sense later, many years later, to retroviruses integrating into eukaryotic host cells as well. And there's a very famous protein, I would say, which I heard as a graduate student, the Lambda C1 Repressor.
Ah, yes.
Which can determine whether the phage will undergo lytic or lysogenic reproduction. And C1 in particular represses the lytic genes and commits it to the lysogenic program. But when it's not there, if C1 is not present, and sometimes it's inactivated by the host stress response, the SOS response of the host, the lytic genes of the phage are produced and the host cell is killed. And you'll hear more about that in this paper. Mitomycin C is used often to induce an SOS response. And I remember that as a graduate student because I knew people working on phages, they'd always adding mitomycin C to induce the phages out of the lysogenic state.
Because it damages DNA, and then the phage see that as a danger signal, and they want to get the heck out of that cell.
Let's get out of here.
Because the ship is going down. It's effectively the abandoned ship signal when you detect damaged DNA by the Rec protein.
So this is the phage detecting stress of the host cell, but there's now some evidence that there are cues beyond the host itself, and we talked about one not too long ago, the Arbitrium system. We talked about it here on TWiM. Some phages encode a peptide, which is detected by prophages in neighboring cells by a receptor that's encoded by the genome. And this phage peptide receptor pair represses lysis. So it's an example of how cells can communicate in some way. And there's some other examples of autoinducers that can regulate viral production. But they say here in the very beginning, the ability of host signaling molecules to double as cues for phage proteins that control fate decisions has never been demonstrated. And that's what they're going to be doing in this paper. So the fate decision is whether you lyse the cell or you become lysogenic. And I learned, which I didn't know, I should know, I guess, that you can be a lysogen not only by integrating, by the phage genome integrating into that of the bacterial cell, but it can remain, in some phages, it remains a plasmid.
Yes, as long as the repressor is made.
That's right. So you don't have to integrate.
Mu is an example.
Yeah, that's right.
And we can think of that as a benign state. It's benign for the bacterial host.
Right.
Even though the term lysogen sounds aggressive, it's not.
All right.
And we can't go on without mentioning the classic book that came out in 1986, The Genetic Switch by Mark Potashny, that describes this remarkable opera that is lysogeny, and he walks the reader through the wonder that is phage lambda. And that book is still in print. It's in its 3rd edition, and you can take a look at that. It's a short book. It's only about 150 pages long, Vincent?
Yeah, I got it right here on my shelf, and you should read it because it's not long and it's really good. It's
cartoon pictures too.
Cartoons, yeah, it's wonderful. Yeah. The Genetic Switch. All right, so this paper starts by looking. So the Vibrio protein, VQMA. This is a protein that binds is a quorum sensor. It binds the DPO molecule, and it also is involved in transcription. So they call it a dual receptor transcription factor. They look at the sequence and say, can we find other proteins that look like this? So they do a bioinformatics search for proteins that look like this sensor in Vibrio. And they come up with 103,000 hits, only one of which was a virus protein.
It's pretty remarkable.
It's amazing. And the virus protein, it's a protein called GP56. And the virus is a myoviridae called VP882, which is a temperate phage, non-integrating, so it remains episomal, and it's from Vibrio parahemolyticus strain. All right, so the phage is called VP882, and it infects a variety of other Vibrios, including...
How far is Vibrio parahemolyticus from Vibrio cholera?
How far is a good question. I don't know. Do you know?
I don't know.
I don't.
You don't know how far it is.
I know both cause food poisoning. Cholera and hemolyticus cause food poisoning, but I don't know genetically how distant they are from one another. And I don't even know if parahemolyticus has 2 chromosomes like Vibrio cholera does.
But
I'm sure we'll get a note from Rita Colwell.
Well, that would be fine.
I can tell you that this particular strain was a long distance from the Bassler's lab Vibrio cholera strain. There had been one paper reporting this lysogenized parahemolyticus Vibrio strain, and it was in Taiwan. And they had a 10-month quest to try to locate the person who might still have a stock of this lysogenized strain. They had great help from a senior scientist, technician, kind of lab administrator in the lab, Jingping Kong, who speaks Chinese. And so she was instrumental in filling out some of the paperwork. They eventually found it was in a stock center in Taiwan. So after a 10-month quest, They were able to track back and then get this strain delivered to the lab. So geographically, they were very far apart. But genetically, I don't know how far apart they are.
I would have just synthesized it from the sequence, although that would have cost more.
Right.
38.2 kilobases. And they had the whole sequence, so they could have done that.
Yeah. And in fact, Justin had...
That wouldn't have given him the bacterial strain.
No, that would have just given him the phage.
Right,
sorry, yes, you're right. You're looking for the bacterium. So this protein, its GC content of the gene encoding it suggests that it is not part of the host. It wasn't transferred from the host. And they noticed that the gene is located between 2 other genes, and the other genes are essential and conserved against across all the phages. And so they thought maybe this protein is important. So they called this. So this is GP56, and afterwards it's going to be called VQMA phage. So remember, VQMA VC is the Vibrio version. So there's a Vibrio sensor of DPO, and as you'll see now, there's a phage sensor, and we haven't yet proven it's a sensor, but we're going to shortly do that. So they take this gene, previously called GP56, now it's a VQMA phage. They clone it into a vector, and they make it inducible with arabinose, and they put it in Vibrio parahemolyticus. And that's the same isolate that Michele's mentioned that they spent 10 months getting, the original isolate in which phage was discovered. And when they induce the production of this VQMA protein, the OD of this culture drops, suggesting it's being lysed.
Mm-hmm.
And so there's just one protein, And now this host, this bacteria, this V. parahemolyticus, of course, has a phage in it. It's got VP882. And if you expose the bacterium to mitomycin C, lysis also. You can take the phage out of the bacterium, you can cure it, and then, of course, if you put mitomycin C in it, it wouldn't kill it anymore. But if you induce the VQMA in that strain lacking the phage, nothing happens. They don't lyse. So it looks like this protein somehow can promote lysis, but it needs the phage around to do that. In addition, when you do mitomycin C induction, not only does the culture die, but it releases phage particles. And if they produce the protein VQMA phage, it also leads to the production of phage particles. So the protein or mitomycin C kills the host, and phage particles are produced. I think VQMA is an unfortunate name. I can't pronounce it very well.
It's a tongue twister, isn't it?
Yes, it is. And plus, you got subscripts, depending on the origin, phage or VC for V. cholera. It's okay. All right. So now we know that in Vibrio, the VQMA binds DPO, the AI, the little molecule, the inducer. And so they say, what if there's no DPO around? You can grow bacteria in the absence of threonine, and then you don't make DPO because it's made from threonine. So that's a nice trick. And if you do that then and produce the VQMA, you don't get lysis. You need DPO. No lysis if you don't have DPO present.
Justin described this as one of those aha moments that we can all remember from our early days in the lab. You come in early in the morning, you open the incubator, and he saw that only when they added DPO did he see clearing of the culture. So there was clearly a major growth defect. Yeah. But if he didn't add DPO, they grew fine. So it was one of those moments where you just feel like, I'm on to something.
Yeah, it's cool.
And one of the things that's the hallmark of quorum sensing is that the inducer, in this case DPO, gets together with the other factor, and you effectively are creating a positive activator that is going to affect a change in the host. So you need the binary. It's not by itself. Mm-hmm. So you need the DPO and you need the other protein product to get together in order to make, if you will, a transcriptional or translational activator, because Vincent hasn't yet told us which one it is.
Mm-hmm. So then they do a series of experiments which show that, in fact, these proteins bind DPO. They show that the phage version binds it as well as the Vibrio
host version.
The host version. So the activity of VQMA phage is mediated by binding directly to the small molecule DPO. There's a lot of work there, but we don't need to go through it. Just take our word that they showed that DPO binds to this protein. So remember, you can express this protein in the bacterium that has the phage in it, and the bacterium will lyse and produce phage. So what is lysing? What is causing the lysis? So they want to know what phage genes are needed. So they did transposon mutagenesis, and they looked at individual phages with insertions in different areas of the genome, and they said which ones, if knocked out by a transposon, will interfere with lysis after inducing VQMA. And they found a gene called GP62. There'll be a lot of genes here, unfortunately.
Oh, yes.
It's hard to keep remembering because sometimes the numbers aren't in themselves. Unless you've been working on it for years, it doesn't mean anything. So mutations with phages with mutations or insertions in GP62 did not lyse after production of VQMA. So GP62 is the one that's needed for lysis. Of course, we don't know how it's working yet, but we will find out in short time. They did a number of very, very cool experiments which show that GP62 acts downstream of VQMA. So you can do a number of in-trans expression experiments to show that if you have activation of this system via VQMA, GP62 is downstream of it. And so their idea is, and this is both for mitomycin C and for addition of DPO. So in other words, either condition, DNA damage or communication, quorum sensing communication mediated by DPO, there are two triggers you need. First, VQMA, and then GP62, and then you get lysis. All right, so what's GP62? They look at it and they say, aha, it looks like Q. And no, Q is not the guy from James Bond movies. It's a lambda protein that looks like a regulator of lytic genes. And so they look around the region of the phage genome encoding. Remember, this is the I forgot the name of the phage already, V88 something phage. They're looking at its sequence. They look at the Q sequence and they say, oh, look, it's right near a 3-gene operon, which encodes GP69, 70, and 71. And the cool thing is the 5' non-coding region has a terminator in it. Okay? It's got a transcriptional terminator. One of the proteins looks like it's got a lysozyme domain, which would be theoretically involved in lysing the host cell. And the operon is not produced in phage that have a transposon insertion in GP62. But it was when 62 is present, you get a lot of transcription. So they're thinking this is probably being regulated by GP62. And in fact, they show that if you make 62, it activates this promoter because they can make a fusion protein of the promoter of the 69 gene, which has the transcription terminator. They can fuse it to luciferase and then measure light. And then there was light, right?
Mm-hmm.
And they can show that 62 allows transcription. So they're thinking 62 is...
Why don't you repeat the point of this experiment?
I will summarize it all. So we found before that GP62 is a gene needed for lysis upon induction of the phage by either quorum sensing or mitomycin C DNA damage. It looks like GP62 is a transcription anti-terminator that allows the synthesis of mRNA encoding lysis genes. So GP62 allows the synthesis of the lysis genes, and they call it Q. So you can forget about GP62. Just remember Q. Q is a protein that allows transcription of lytic genes. Okay.
And that is from Lambda. Q is from Lambda, and
Right.
it's the simple take-home is GP62 is predicted to encode a protein similar to Q from phage Lambda.
Yeah, that's why this is all based on Lambda work, because they can look at the proteins and go, oh, it looks like Q, and you'll see more. More is coming. All right, so Q is an anti-terminator that, when present, allows transcriptions and synthesis of proteins involved in lysis. So how do you regulate Q? Because if you don't regulate Q, you're going to always have lytic replication. So they made a fusion of the promoter for Q with luciferase. It's called PQ Lux. So you can measure light. And if you take this Vibrio strain without the phage, you make a lot of light from this promoter construct compared to the strain with the phage in it. So somewhere in the prophage, there's a repressor of the promoter for Q.
Mm-hmm.
And that promotes lysogeny. So they look and they say, there's an open reading frame right near here, right near this promoter that encodes a DNA binding repressor. And so they produce this protein and they show it can repress the promoter of Q using the luciferase reporter. So and it's called GP59. It is the repressor of lysis. Prevents somehow the Q from anti-terminating the lysis genes. Now, more Qs from Lambda. The C1 repressor is cleaved upon stress, induction of stress, the SOS response. So they said, maybe GP59 is cleaved also. So they make some fusion proteins and look at that, and indeed it is cleaved in mitomycin C-treated cells. That cleavage depends on Rec-A, it's a DNA recombination protein. And it's the same so they changed the name of GP59 to C1.
Mm-hmm.
Again, it's like a homolog of all these lambda proteins. They're just confirming the same activities. So C1 is a lysis repressor, formerly GP59. We'll summarize all of this in the end. But it's hard. I know it's hard to follow because I had to read this paper many times to keep it straight in my mind. So far, C1 represses the gene encoding Q, which is an anti-terminator. C1 is cleaved on DNA damage, and that launches the lysis program. If you add DPO, which is sensed by VQMA, it also launches the lysis program. So they want to connect these two activities. How can you do that? So first, they have a lysogen of this VP, Vibrio parahemolyticus. It's got a plasmid with a reporter for Q, PQ lux, and PGP69 lux, which is the target for Q. You add mitomycin C to this culture, you get PQ lux first activated, and then 10 minutes later, PGP69. And they say this offset of timing is consistent with the idea that Q is produced first, and then later the lysis genes are made because Q is an anti-terminator. So that's with mitomycin C. They do the same experiment with quorum sensing. They induce VQMA, and they see this 10-minute lag. So they say, this is the same lag, and therefore these two proteins are both involved in DNA damage and quorum sensing, launching of the lytic cycle. Are we okay so far?
We're okay.
Yeah.
Elio?
Yes. A tentative yes.
All right. Then they say, how does this VQMA? Remember, VQMA from the phage is binding the quorum, the AI, that little chemical.
The DPO.
DPO, thank you. So how does that work? How does it initiate the lysis program? Okay, there are a couple of different possibilities, but the one that they show is correct is that there's a third component that's involved. There's another element in the phage, and they should do some experiments showing it's encoded in the phage genome to connect VQMA bound to the compound with C1 expression. And to do this, they take VQMA phage, they put it into E. coli. It's arabinose inducible. They have PQ lux and C1 repressor. And into this, they induce, so that alone will not lyse with mitomycin C or the little compound. So then they add fragments of the phage genome, DNA fragments, and ask for one that restore lux expression following induction. I said lysis, but it's actually just light.
They're just measuring light.
I thought that was a cool functional screen. Let's just chop up the phage genome and see which fragment gives us activity, and then we can focus on that piece of DNA, figure out what it encodes.
So they found a fragment, a single 600 base pair fragment that gave light. It encodes one protein called GP55, and they showed by electrophoretic mobility shift assays that it binds phage DNA that the VQMA binds the DNA just upstream of the GP55 open reading frame. So
when it has DPO attached.
When it has DPO attached to it, but that's the target, and that's linking,
yes,
VQMA to C1. And that's they do in the next experiment, they link GP55, VQMA, and C1 expression. And they show that induction of GP55 is sufficient to turn on PQ lux in this strain. And if you have the lysogen in there, it will cause lysis. So GP55 connects this quorum sensing to derepression of Q, leading to cell lysis. Now, what is GP55? Now, here is an interesting story. It's a small protein, 79 amino acids, with no domains and no significant homology to any protein in the database.
I hate when that happens.
Nevertheless, so they say,
but it gets you into cell.
It does. It does get you into cell. And they say, well, maybe it's acting post-translationally. And they showed that, in fact, it will bind GP55 when made and purified, will bind C1. So basically, it's an anti-repressor that will prevent C1 from binding to Q promoter DNA. So,
and what's cool is they didn't just do this biochemically, but they also expressed tagged versions of each of these components, the Qtip anti-repressor and the C1 repressor, in the same cell. And they found that the Qtip caused dramatic redistribution of the C1 repressor, which normally is filling the cell cytoplasm, and it sends it instead to the poles. So Justin said the day he looked in the microscope and saw that dramatic difference was another really exciting day, because, again, he had just screened the whole genome, found this fragmented DNA, and then to be able to go a few steps further and see that it is causing this dramatic relocalization of a target protein in whole live cells was really, really proof that he's onto an important molecule.
And you don't need to be a skilled confocal microscopist to appreciate that the protein is indeed moving. I mean, it's like you colored it on with a colored pencil or marker. On
this is figure 4D in particular.
Yes. I mean, it's just remarkable.
So you may have noticed that they renamed GP55 Qtip for quorum-triggered inactivator of C1 protein. I bet they like that name, Qtip, right?
I think they went to Qtip and then figured out all the words to go with it.
Well, they too were struggling, I'm sure, with all this GP59, GP62, GP. So it helps us, and I'm sure them too, to communicate with their lab members if they can simplify the terminology a bit.
The aggregation is interesting because Qtip aggregates C1, and in contrast, when you treat cells with mitomycin C, C1 just gets cleaved and it doesn't aggregate at all. So there are two different mechanisms of sequestering or inactivating C1. There's the Qtip one, and then there's the one by the host protease. Was it the LON protease that does that, Michael?
I think you're right.
Gosh, some of the old molecular biology is coming back.
All the neurons are firing today.
There was a reason I took all those courses as a grad student. Yeah.
Well, like I said, if you get stumped reading this paper, pull out your copy of the Genetic Switch, give it a quick skim before you attack this paper, and this paper will then become much easier to get through if you remember your lambda biology.
And their graphical abstract at the start of the paper is also a great guide.
Yeah, that's really it. That's the summary, They looked in so this is a new thing, this Qtip, and they say it hasn't been reported before. So they looked in some other databases, and they found an example in a Marinobacterium, which had some similar proteins in it. And that bacterium has a phage in it that has a C1-like gene, and Qtip could actually sequester it as well, but not others. And they ended up looking at 9 other linear plasmid-like phages. And in 5 of these, they could find in this region that encodes Qtip VQMA in their phage, they could see small genes and oppositely oriented ORFs, which is what's happening here with the Qtip and VQM with DNA binding domains. So they said there are probably other phage-coded DNA binding proteins that work in a similar way. And they're just different proteins, so we're not recognizing them.
Mm-hmm.
And if you think about it, this particular phage organism cohort would want to be able to suppress lysis because it's an organism whose normal niche is water. Mm-hmm. It's not living where it's likely to encounter another viable host if the host it's living in should lyse, secondary to whatever happens to trigger it. So you're going to want to put that extra level of control in order to preserve your genes to go to another generation.
Mm-hmm.
And it makes sense if you think about the life history of Vibrio cholera in the fact that it's living in water as opposed to a niche or as a facultative intracellular parasite like it does in the copepods that Vibrio normally inhabit in water when it's effectively a different niche.
Mm. So then they say, can we put this system into Vibrio cholerae? And what happens? So they take VQMA phage, and Vibrio cholera has its own VQMA, VC, of course. They put the whole prophage in Vibrio, and they do a series of experiments. And the bottom line here is that the phage VQM can substitute for the Vibrio protein, but not vice versa.
Oh,
and that's interesting. That's a very interesting observation, which we'll come back to later after we get some more information here. And they also show that even in Vibrio cholera, the switch, the lysis lysogeny switch, can be regulated again by DNA damage or by the DPO binding to VQM. In this case, if you put the phage VQM, it works as well. And it is another series of experiments. They show that it is the host synthesized DPO that is sensed by the phage VQMA protein that determines the lysis lysogeny switch. Okay, and it works through Qtip and C1 sequestration, as we said before. So input from Qtip enables the phage decision, lysis lysogeny, and that, of course, is connected to cell density, because as the cell culture density increases, it makes more DPO. That's sensed by the phage protein, and that can then start this cascade leading to the lytic cycle. And then just a few more interesting experiments left. They say, can we manipulate this system to make it a V cholera-specific phage kill switch? And so they take a Vibrio-specific promoter and they put together the system where, in the presence of DPO, and this is prophage, of course, the phage will go into the lytic cycle and kill Vibrio cholera specifically, but not parahemolyticus or vulnificus, because they use a promoter that's only recognized in Vibrio cholera to drive the system. So that's cool, because now you could think about designing such kill switch systems for specific bacteria. And they extend it to Salmonella typhimurium, where they use a Salmonella promoter. Same idea, and they show that you can kill Salmonella specifically by this Quorum Sensing system using the phage-encoded sensor protein. And so you can imagine that this may have some broad use if you want to design phages that are very specific for certain bacteria. They actually say we can imagine programming phages to contain different logic circuits and multiple input dependencies.
Wow.
Thinking about that engineering, it may not surprise you to learn that Justin's undergraduate and graduate, or he got a master's degree, was at a College of Engineering here at the University of Michigan. So he has this really solid background in engineering principles that he then applied to this really cool biology.
That's neat. Now, so they say this is the first example of a phage lysis decision made by a phage-encoded receptor that monitors an AI produced by the host. But they say there are probably other examples out there. And this idea that the phage protein can substitute the VQM can work for that of the host, but not vice versa. They say this could mean that the phage can respond to the host, but maintain its ability to do its own thing without host interference. And that's a cool idea, right? So the phage could respond to the host density, but the host is not going to control the phage because its protein doesn't work for the phage. And I think that's pretty cool. The other thing,
and
go ahead.
Miraculously, they reduced the entire manuscript to one sentence simply by saying bacteriophages listen in on their hosts.
They listen in.
Yeah.
That's beautiful. That's really nice.
And not just the individual cell, but now how many
the community
cells are nearby. Yeah.
Right.
I thought it's really elegant.
So let me just summarize it before Michele tells us a little more about Justin. So
Sure.
You have low cell density, low amount of autoinducer, in this case, DPO. When the cell density gets high, it makes more DPO. DPO is sensed by the phage sensor protein, turns on the synthesis of Qtip, and then Qtip sequesters C1 into these aggregates, and that allows turning on of the lytic genes and lysis. So in sensing, as Michael said, it's listening in, and it can sense when's a good time to start replicating when there are lots of hosts around.
So your genes go to the next generation.
That's right. That's right. Anyway, it's a pretty elegant series of nice experiments, and
It's really lovely.
Working on, really, it's building on the shoulders of giants, right?
Mm-hmm.
Right. And as you were pointing out, they see hints in other genomes that a similar system could be at work. And so that's one of the things that Justin is pursuing now, to see if this same architecture or circuitry, rather, is being used for other phage to kind of sense different environmental cues, not just classical DNA damage, for example. So Justin Vincent was born in New York City, and then he grew up in Westchester County, but came to University of Michigan for his undergraduate work and was enrolled in a 5-year program called Concurrent Undergraduate Graduate Studies Programs at the College of Engineering, which you get both an undergrad degree and a master's degree. And it provided opportunity for him to do research not only at Michigan, but also he ended up collaborating with a group at Princeton and actually did about 9 months of research at Princeton during this 5-year undergrad and master's program. And that's how he first learned of Bonnie Bassler and her work. So he really appreciated the exposure to laboratory science. He was in a lab in the chemistry department here, and he said a summer fellowship is when he was working full-time, and science really took hold of me. He fell in love with research and wanted more. And because of this collaboration with a colleague at Princeton and his time there, he became familiar with Bonnie Bassler's work. So when he applied to Princeton, his goal was to see if he couldn't study under her for his thesis. But he says what really sealed the deal is during his rotation. So typically in a graduate program, you rotate through maybe 3 different labs and get some exposure to the type of thinking that they do and the culture of the lab. He was teamed up with a postdoc, Kai Pappenfort, who he really credits with teaching him how to be a scientist and instilling in him the passion for rigorous research. So it was with Kai that they first discovered this DPO quorum sensing molecule and also its receptor. And they published that together in a Nature Chemical Biology paper. And then Kai went on. He's now a professor in Munich in Germany. But then Justin took the lead and did the beautiful work that we heard about today. I've already described some of the really exciting moments in the lab, but as he reflects back on his experience in Bonnie's lab, he, first of all, is appreciative for the patience that the other lab members showed when he was presenting his early work in lab meetings and, ran into some real puzzles and really appreciated some ideas and patience that the lab members showed. But he was especially grateful for Kai Pappenfort, who was really a role model and inspiration for him. I asked him if he had advice for junior scientists, and he again thought of Kai and said, you really want to be surrounded by people who are passionate about what they do, that are excited, that will think hard about your work and make you realize that it's important and that you're doing something of value. So that's his advice. He does remember that his mind had to be completely rewired, came again out of University of Michigan with an engineering training, and now he was stepping into this world of biology. So he really appreciated that the Bassler lab encourages that interdisciplinary research, and he's grateful that he did incorporate biology into his portfolio, especially some of those microscopy experiments that we talked about. Right now, he is exploring whether a similar circuitry is operating in other phage host systems. He hasn't yet decided what he'll do next, but he met another Michigan grad when he was here. She is now his fiance. Katie Wood is working at IBM. She's also a Michigan native, and so the two of them will be launching the next stage of their promising lives together. And I'll close by just saying that I think this is the first time in TWiM history where we have featured Michigan in back-to-back presentations, both the snippet and the paper. So I'm feeling great about the new year.
Fungus and an AI.
Yeah.
And here I thought you were going to say when he left Michigan to go to Princeton, he went from a winning football team to a losing football team.
It's basketball season, Michael.
I know.
And I'm going to leave it at that.
If you had trouble with today's discussion. There is a blog post I can recommend over at Virology blog. I had a guest blogger, Trudy Ray, write a post about this article. It's called "These Bacteria Are Bugged," and I will put a link to that in the show notes.
That's
if you
cute.
Want to check it out. Yep.
That could be a cool title for this one.
Yeah, well, I don't want to steal her title. We'll come up with something else.
Yeah.
All right. That's TWiM 192, first for 2019, first of many. You can find it at asm.org/TWiM. If you haven't been to asm.org lately, check it out. It's newly redesigned. It's very cool. And there you will find show notes and so forth. And send us your questions and comments to twim@microbe.tv. If you listen on your phone or tablet, please subscribe on your app that you use. It really helps us to know how many people are listening. And also, if you really like what we do, consider donating financially. You can go to microbe.tv/contribute. We have a number of ways you can help us out, including $1 a month over at Patreon. Today's TWiM has been Michele Swanson from the University of Michigan. Thank you, Michele.
Thank you. It's a pleasure.
Elio Schaechter from Small Things Considered. Thank you, Elio.
My pleasure again. Sure.
Michael Schmidt from the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.